Hi, my name is Mrna, and I'm going to be presenting my video on gene expression and regulation. So first, let's take a look at these two images. So on the left side, we have a girl who is very happy and smiling, and on the right side, we have chimpanzees. So obviously, this human girl right here is very different from chimpanzees. But what if I actually told you that humans and chimpanzees have DNA that is actually about 99%? That is about 99% similar. And you might be wondering, whoa, how? Well, that's due to gene regulation. So let's talk about eukaryotic gene regulation and then prokaryotic gene regulation. Remember, eukaryotes have more than one cell, such as humans and animals, and prokaryotes just have one cell, such as bacteria. In the human cell, there are many different types of cells, ranging from liver cells to kidney cells to fat cells. Almost all of these different types of cells in the body contain the exact same set of DNA instructions. Then how do they look so different and carry out such different tasks? This is due to gene regulation. A quick overview. So first let's talk about these proteins. So proteins, they carry out these wide range of functions in cells, and they're very important for organisms to live. They maintain homeostasis, and they're important for growth and tissue repair. So they're very important. But in order to make them, we need to go through this process called transcription and translation. So DNA has the instructions that code for the protein, and however, it's in the nucleus, so it can't always leave it all the time, and it can't leave the nucleus. So what we need to do is we make this thing called an mRNA using this process of transcription. So RNA polymerase and many other enzymes involved to make this pre-mRNA, and then it is processed into mRNA. So using this DNA strand, we make a complementary RNA strand, and then we process and make this mRNA, and we make this like messenger RNA, and then this message is, and then we go to this process called translation, and then this message is used and it is translated in the cytoplasm. So here we were in the nucleus, then we carry on over to the cytoplasm. And then in the cytoplasm we have ribosomes. This is where translation occurs. For ribosomes are the site of protein synthesis. So we translate that message, we have into amino acids, and then all these amino acids are joined together and we make polypeptides or proteins. So, what is gene regulation? Gene regulation is the process by which a cell controls what genes to turn on or express out of the many different genes the cell has in its genome. Because of gene regulation, each cell type in the human body expresses a different set of genes, even though they all have the exact same set of DNA. Exact same DNA. The cells turn out to be different because they have different patterns of gene expression, and this causes them to have different sets of proteins. This is the reason that these cell types have unique functions compared to one another. Remember, these proteins carry out these wide range of functions. So how do these cells decide what genes they want to express? A cell's gene expression pattern depends on both the cell's internal state and external state. In other words, the gene expression pattern depends on information outside and inside the cell. Examples from outside the cell includes like chemical signals from other cells, mechanical signals from the extracellular matrix, and nutrient levels. Inside the cells, examples from inside the cells, the proteins it inherited from its mother cell, whether the DNA is damaged or not, and the amount of available ATP. ATP is basically just energy. How do these cells use this information to decide what genes to express? Well, cells have molecular pathways which convert information. For example, the binding of a ligand to a protein receptor, which results in a change in gene expression. So this right here is a molecular pathway. It's a signal transduction pathway. So we have like this signal, and it binds to this receptor. We go through all these different processes, and then at the end we have cell growth and division because of this one signal. So the regulation of eukaryotic gene expression. The expression of genes in a eukaryotic cell can be regulated at different stages. A single cell can even be regulated at multiple stages, and the main stages are the following. And we'll discuss this in the coming slide. So the explanation of the stages. Chromatin accessibility. The structure of the chromatin can be regulated. So chromatin is just basically DNA, and it's organizing proteins. More loose or open chromatin allows for a gene to be more available for transcription compared to a more tightly coiled chromatin, which makes it harder for that same gene to be transcribed. So right here, if it's more open, easier for the protein to be made. Transcription. This is the main regulatory stage in eukaryotic gene regulation. In transcription, remember, we're making the mRNA. Transcription factor proteins bind to specific DNA sequences near a gene, and they either promote or repress its transcription into an RNA. RNA processing. Splicing, capping, and addition of a poly A tail are these processes in RNA processing. These can be regulated, and so can the exit from the nucleus. Different mRNAs can be made from the same pre-mRNA using alternative splicing. This is due to the structure, because of the exons. You can use introns as exons in other types when you're making other types of proteins from the same mRNA. So RNA stability, the lifetime of an mRNA molecule in the cytosol affects how many proteins can be made from it. Small regulatory RNAs called miRNAs bind to target mRNAs and cause them to be chopped up. Translation of an mRNA may be increased or inhibited by regulators. For example, miRNAs sometimes block translation of their target mRNAs rather than causing them to be chopped up. Protein activity. Proteins can undergo a variety of modifications, such as being chopped up or tagged with chemical groups. These modifications can be regulated and may affect the activity or behavior of the protein. So now let's talk about prokaryotic gene expression. Remember, prokaryotes just have one single cell, and this gene expression in these organisms is not as complicated as it is in eukaryotes. This is because they have much less DNA, and they use operons for groups of related genes. Operons function as simple on and off switches. So let's talk about the operon. In prokaryotic organisms, whether a gene is transcribed is dependent on activators and the repressors that are present. So what are operons? Operons are a region of DNA with the following parts: a promoter, an operator, and one or more genes which code for a protein. And so repressors bind to the operon to stop a protein from being made, and activators bind to the operon to help make a protein. So let's take a look at a real-life application of this prokaryotic gene regulation. So let's take a look at the Lac Operon, which is used in bacteria to break down lactose when there's too much lactose. So the promoter is the region of the DNA where the RNA polymerase binds to. An operator is like an on and off switch, which turns the operon on and off, and the repressor turns off the expression of a gene. So how does this work? So when there's too much lactose, what it does is it binds to this repressor, as we can see here. And this causes a conformational change in the repressor, and it falls off because it changes shape from the operator. And when that happens, this repressor is no longer blocking RNA polymerase from making this mRNA. Remember, in transcription, we had the mRNA, and RNA polymerase is one of the enzymes that helps make this mRNA, and then we can make a protein from that messenger RNA in translation. And once we make these proteins, they're enzymes that eat up this lactose, they break it down. And when all of this is gone, this lactose right here, it's going to be gone. And then this repressor can bind back to the operator, blocking RNA polymerase. And then we won't need these proteins anymore because we don't have any more lactose since the enzymes already broke it down. So, were you paying attention? In which of the following stages can gene expression be regulated? A. Transcription, B. Translation, C. Chromatin accessibility, and D. All of the above. I'll give you a second to think about it. If you press D, good job, that's the right answer. So here are the sources used in this video, and that is the end. I hope you enjoyed.